Hello viewers. First of all, I would like to give my sincere thanks to the Zoological Society of Assam for taking up such initiatives to help the students during this pandemic period. This presentation is on the topic T-cell receptor, picked up from the PG syllabus of Guwahati University. The learning outcomes of this presentation are T-cell and its types, major histocompatibility complex and its types, T-cell receptor model, T-cell receptor and its types, T-cell production, maturation, and generation of T-cell receptor, selection process of T-cell, structure of T-cell receptor alpha beta, CD3 complex, structure of CD3 complex. Before coming to the actual topic, we have to know about the T-cell because T-cell receptor is present on the surface of the T-cell. So what is a T-cell? T-cell, which is a type of lymphocytes or white blood cells derived from the lymphoid progenitor cells during hematopoiesis in bone marrow and has undergone the maturation process and thymic selection and mainly involved in adaptive immunity. There are mainly 4 types of T-cells: helper T-cell, cytotoxic T-cell, memory T-cell, and suppressor T-cell. Let us know a little bit about these cells because some terms like major histocompatibility complex associated with these cells will be required in the later slides. So I will try to present these terms one by one. Now, the helper T cells, who helps the other cells of the immune system to eliminate the pathogen. But before that, they have to activate themselves, and this is done when they are presented with the peptide antigens or foreign invaders by major histocompatibility class 2 molecule. These major histocompatibility class 2 molecules interact with a protein called CD4+, which is present on the surface of the helper T cells, and due to this, helper T cell is also known as CD4+ cells. Now, what is major histocompatibility complex and CD4+? CD4+, that is cluster of differentiation 4, which is a glycoprotein found on the surface of the immune cells like T helper cells, monocytes, macrophages, and dendritic cells. So this is a CD4 glycoprotein, and on the surface of the T helper cell, CD4+ is present. Now I will come to this MHC molecule in another slides. Let's first talk about the other T cell types. Cytotoxic T cell. They are the soldiers of our body because they kill their target cell directly. They are licensed to kill, to destroy. This cell recognize their specific antigen when presented by MHC class 1 molecule, and on the membrane of their surface, CD8+ glycoprotein is present. So we can distinguish T helper and T cytotoxic T cells from one another by the presence of either CD4+ or CD8+ membrane glycoprotein on their surfaces. Memory T cell. As we all know, following an infection, antigen-specific long-lived memory T cells are formed. They are important because they can quickly expand to large amount of effector T cells upon re-exposure to the antigen. These memory T cells can either be CD4+ or CD8+. Regulatory T cell, formerly known as suppressor T cells, help damping the activity of T cells by modulating the immune system, by maintaining tolerance to self-antigens, and prevent autoimmune disease. So I have discussed recently about the cytotoxic T cell, like they are the soldiers of our system. They went to attack our enemy, they taken care of it. Now we need to snatch the power from them. So how does regulatory T cells do it? They do it by inhibiting T cell proliferation and cytokine production to dampen or to suppress or to regulate the process. That is why these cells are called regulatory T cells. Now, what is the major histocompatibility complex? Major histocompatibility molecules are so named because they are first identified as antigens responsible for the acceptance or rejection of tissue grafts upon transplantation. They are the collection of genes present on the chromosome number 6 in human and 17 in mice. MHC plays a pivotal role in the adaptive branch of immune system. They present the peptide antigens derived from cell protein or from pathogen and present them on the surface for recognition by the appropriate T cell. One thing I would like to mention that T-cell receptor that are present on the surface of the T-cell cannot recognize an antigen until and unless the antigens are presented by the MHC molecule. There are three classes of MHC protein. Class 1 MHC genes are expressed on nearly all nucleated cells. Their main function is to present the peptide antigen to cytotoxic T cells. Class 2 MHC genes are expressed only by antigen presenting cells to the T helper cells. The antigen presenting cells are macrophages, dendritic cells and B cells. Class 3 MHC genes helps in the components of the complement system. The search for the T-cell receptor started way back in 1976 when R.N. Zinkernagel and P.C. Doherty in 1974. Their studies demonstrated that antigen recognition by T-cell is specific not for viral antigen alone but for specific antigen associated with an MHC molecule. They shown that T-cell recognizes antigen when they are presented on the membrane of a cell by a self-MHC molecule. This attribute is called self-MHC restriction. For this work, Zinkernagel and Doherty awarded Nobel Prize in 1996. This led to the proposal of two models, dual receptor model and altered self model. So, what is a dual receptor model? I will try to present it by a diagram. Dual receptor model predicts that T-cell receptor has two receptors where one receptor recognizes the antigen present on the cell surface and other recognizes the MHC molecules located at some distance. From this we can understand that antigen and MHC molecules are recognized separately and that too by separate receptors, like this one is receptor and this one is another receptor. They are both separated. Two receptors are there. Altered self model suggested that T cell has only one receptor and that is designed to recognize both the components antigen and MHC molecules. So here this is the one receptor, only one receptor is present. The debate between proponents of these two models was carried on for a number of years until an elegant experiment by J. Kepler and P. Marek demonstrated that specificity for both MHC and antigen resides in a single receptor. An overwhelming amount of structural and functional data has since been added in support of the altered self model. Now, how do we distinguish a T cell? Simply by the presence of a T cell receptor on its surface. So it is found on the surface of the T cell. There are about 20,000 receptors molecule on the surface of the T cell. The main function of the T-cell receptor is to recognize antigens which are presented by major histocompatibility complex molecule. There are two types of T-cell receptor. One is TCR alpha beta, another is TCR gamma delta. The majority of the T cells in human expresses TCR alpha beta, about 95%, and 5% by TCR gamma delta. T cell express either alpha beta or gamma delta but not the both. Before starting the generation of T cell receptor, we have to know about the T cell production because the generation of T cell occurs simultaneously with the production of T cell. T cell production occurs in bone marrow. In bone marrow, hematopoietic stem cell give rise to the common myeloid progenitor and common lymphoid progenitor cells. This common lymphoid progenitor cell give rise to the T lymphocyte, B lymphocyte and natural killer cells. So I will talk about only the T lymphocytes. Now these common lymphoid progenitor cells migrate as immature precursor T cells via bloodstream to thymus gland. In thymus gland, now they will be called as thymocytes. These thymocytes go through a series of maturation steps and they become the naive T cell and migrate to the peripheral tissue. These naive T cells are innocent because this has not encountered any antigen till now. So what is happening inside this thymus? I will discuss it in the next slide. That is T-cell maturation and generation of T-cell receptor. Because I already have mentioned that T-cell receptor generation occurs simultaneously with the maturation of T-cell. This is the cross-section of thymus. The precursor T-cell now arrive at the subcapsular cortex region of the thymus. From the subcapsular cortex, the cells migrate to the cortex where the rest of the maturation events takes place. Once in the cortex, these cells undergo high mitotic activity to form a population of large proliferating cells. Generation of large pool of cells is necessary because a larger number of antigen-specific T lymphocytes needs to form that ultimately recognizes a large variety of antigens. The first stage of thymocyte is double negative. So, what is this double negative? In the previous slide, I have mentioned about the types of T cells, that helper T cells, which is also called as CD4 plus cells, and cytotoxic T cells, which is also called as CD8 plus cells. So double negative meaning this CD4 and CD8 are absent on the surface of this particular thymocytes. Double means CD4 and CD8 plus, and negative refers absent, hence double negative. Another protein I would like to mention is CD3, that is cluster of differentiation 3, which is involved in activating both the CD8 and CD plus cells. The CD3 protein is also not expressed in the outside. They are still in the inside of the cell. Now we term this cell as pro-T cell double negative. And till now, T-cell receptor is also not expressed. Because till now, these progenitor cells have not arranged their TCR genes and expression of the protein is yet to be done. The proteins are recombination activating gene, that is RAG. Specifically, RAG1 and RAG2 are required for the rearrangement of the T-cell receptor genes through VDJ recombination. So, VDJ recombination is a process that occurs in the early stages of T-cell and B-cell maturation. T-cell receptor needs to generate the diversity because there are billions of pathogenic antigens, and the main job of the T-cell receptor is to recognize them. Therefore, the diversity of T-cell receptor is needed. The next step is, now this pro-T cell becomes the pre-T cell. In this stage, the thymocytes stop proliferating and starting rearranging their TCR genes, that is T-cell receptor genes, and express CD3 on their surface. The first set of rearrangement occur as the T-cell receptor beta, gamma and delta genes rearranged. If the T-cell makes productive gamma and delta change in rearrangement, it can express the TCR gamma delta receptor. The functional gene arrangement of the beta chain locus occurs prior to the alpha chain. If the cell make a productive beta chain gene rearrangement, it makes a pre-T cell receptor that consists of beta chain and pre-T cell receptor alpha chain. Cells that neither produce alpha beta T cell receptor nor the gamma delta receptor usually die at this stage of development. Now this pre-T cell advance to the double positive stage, meaning both CD4 and CD8 are expressed on the cell surface. TCR alpha chain expression does not occur until the cells are double positive. So once the thymocytes are double positive, they stop dividing and increases the level of the recombination specific RAG2 and RAG1 recombinase, which induces alpha chain rearrangement. Once the alpha chain rearranges, the T cell expresses complete alpha beta cell T cell receptor and CD3 complex on its surface. Now these double positive cells become responsive to antigen and are subjected to positive and negative selection, and after that they become CD8 plus T cells and CD4 plus T cells. I have to talk about a little bit of the selection process, otherwise the whole process will be incomplete. The selection process is also called the thymic selection. The large number of immature T lymphocytes, that is double positive cells, undergo positive and negative selection before they mature into CD4 plus and CD4A T cells. Positive selection takes place in the cortical region of the thymus and involves the interaction of immature thymocytes with cortical epithelial cell. This is the thymic epithelial cell with self-MHC plus self-peptides. Positive selection selects only those double positive thymocytes that express T-cell receptor which binds with self-MHC plus self-peptide complex with low affinity. That is, they become self-MHC restricted, meaning they will not fight with our own MHC, our own self-peptide. Now these thymocytes are permitted to survive by the epithelial cell by sending a signal that switches off apoptotic machinery in the thymocytes. The thymocytes whose TCR are not MHC restricted, that if they do not recognize major histocompatibility complex, will die. Because the positive selection instruct or teaches a population of thymocytes to recognize only self-MHC associated with self-antigen or peptides. So what is the result of the positive selection? The result is the T cell becomes self-MHC restricted. Negative selection, which takes place in the medullary region of the thymus. The medullary dendritic cells and macrophages expresses class 1 and class 2 MHC molecules with thymocytes. This is the CD4 and CD8 thymocytes exposed with a self-MHC plus self-peptides. This bind with low affinity and this bind with high affinity. Negative selection eliminates those T cells that react too strongly with self-MHC plus self-peptides, hence allowing the maturation, allowing the T cell which have weak affinity for self-MHC plus self-antigen. So what is the result of the negative selection? The result is development of tolerance, that is tolerance to self-antigen. Now let us know about the structure of the T-cell receptor alpha beta. T-cell receptor alpha beta is composed of alpha chain and beta chain. Each chain has two extracellular domain, outermost variable domain V alpha and constant domain C alpha. Each domain is stabilized by an internal disulfide bond. In addition to the constant domain, a short connecting sequence where a cysteine residue forms a disulfide link with the other chains of the heterodimer, that is alpha and beta. Transmembrane region anchors each chain in the plasma membrane. Each T-cell receptor has a cytoplasmic tail of about 5 to 12 amino acid at the carboxyl end. The variable domain of both chain together form antigen binding site. Because the job of the T-cell receptor is recognize an antigen when presented by MHC molecule, and alpha and beta chain do this job. After the T-cell receptor recognize the antigen presented by the MHC molecule, the signal is transmitted from the surface nucleus, which leads to the activation of the T-cell. But one problem encountered: the T-cell cannot do this alone because the cytoplasmic tail of T-cell receptor is extremely short, that is 5 to 12 amino acid long. Therefore, for a signaling purpose, we need another complex of protein called CD3 or cluster of differentiation, which is associated non-covalently with a 5 transmembrane protein called cluster of differentiation 3. These 5 proteins are associated to form 3 types of dimers: dimer of gamma and epsilon chain, dimer of delta and epsilon chain, and a homodimer of 2 zeta chains, or a heterodimer of zeta and eta chains. What is the role of CD3 protein? It has a role in membrane expression of T-cell receptor as well as the signal transduction inside the cells, which results in the T-cell activation after the T-cell receptor binds with the antigen MHC complex. Together, the T-cell receptor with CD3 protein is called T-cell receptor complex or CD3 complex. Let us have a look on the constituting polypeptide of CD3 complex. Here, the protein TCR alpha and TCR beta recognizes antigen plus MHC, and the size is 40 to 60 kilodalton and the size of TCR beta is 38 to 50 kilodalton. TCR gamma and delta recognizes antigen. The size of TCR gamma is 40 to 55 and TCR delta is 40 to 60 kilodalton. The CD3 gamma, delta, epsilon, zeta and eta, all the functions of all these proteins are signal transduction, and the size of the CD3 gamma is 25 to 28, delta 20 to 28, epsilon 20 to 25, CD3 zeta is 16 kilodalton and CD3 eta is 21 kilodalton. Let's have a look at the structure of this CD3 complex. This is the alpha and beta chain of the T-cell receptor complex. The gamma chain of CD3 complex contains N-terminal extracellular domain followed by a transmembrane region containing negatively charged aspartic acid group. This is the transmembrane region. The transmembrane region is followed by a cytoplasmic tail about 44 to 81 amino acid residues. This is the cytoplasmic region, all this region. The cytoplasmic tail of this gamma chain contains one pair of motif called the ITAM, immunoreceptor tyrosine based activation motif, which play a role in signal transduction. The alpha chain has the N-terminal immunoglobulin like extracellular domain, a transmembrane segment containing negatively charged aspartic acid, and a cytoplasmic tail containing a single ITM sequence, and together this gamma and epsilon, together with these two domain, together all the structure, we called it as a gamma and epsilon chain. So like its sister chain gamma, delta chain has N-terminal immunoglobulin like extracellular domain, a transmembrane region containing a negatively charged amino acid, and a cytoplasmic tail containing a single ITM motif. And this is the epsilon chain that I already have talked about. So together they make the delta epsilon chain. Zeta chain is a 10 kilodalton non-glycosylated protein which has no sequence or structural homology to gamma, delta, epsilon chains of CD3 complex. This exists as a homodimer interlinked by a disulfide bond. This is the disulfide bond. It has a very short extracellular domain of 6 to 9 amino acid. The transmembrane region has a negatively charged amino acid, and a cytoplasmic region contains 3 ITM. So this is all about the T-cell receptor. With this, I have come to an end of this presentation. Here I have provide references with suggested readings which viewers can go through. Thank you all to the viewers. If you have any feedback or any kind of queries, please feel free to contact me. Here I have provide my email ID floranayak@gmail.com. Thank you.